No. Hey everyone, this is going to be the second component of the protein structure lecture. And so I'm going to, it's okay. I just got some strange notification on my computer, so disregard that. So we're still in chapter number 3 and 4. It's kind of a blending of the end of chapter 3 with the beginning of chapter 4. I'm going to fill in some of the gaps and refer back to some of the things that I mentioned whenever we were talking about hemoglobin and its structure. But we'll do that as we encounter them. So here's your list of learning objectives. There's some things about different types of interactions. Those are definitely a big part of this chapter, non-covalent interactions, covalent bonds, your different structures, levels of structure, primary, secondary, quaternary structures. And then there's some items about P50, so hemoglobin's affinity for O2. And your P50 is the amount or the concentration of O2 required for your protein to be 50% saturated. Okay, so we'll proceed. There's also some comments about the T state and the R state. When we were talking about hemoglobin, the T state was a deoxygenated form of hemoglobin. R state was our oxygenated state of hemoglobin. Now, with that said, there are some very important key properties of proteins. Proteins are that they are linear polymers composed of amino acids. Proteins have a very wide variety of functional groups. Proteins can interact with one another and other macromolecules to form complexes. Now, this is an important factor from a structural standpoint. Some proteins are very rigid, while others are very flexible. And to illustrate, now there's a total of 4 different levels of protein structure: the primary, secondary, tertiary, quaternary structure. Primary, secondary, tertiary, quaternary. Now, to illustrate these, and I think an important point that I've made in one of the other videos was how these individual amino acids play into these different levels of structure and what stabilizes these different structures. Your primary structure is stabilized by peptide bonds. Your secondary structure is stabilized by backbone, H bonds, hydrogen bonds. Your tertiary structure is stabilized by R group interactions. But one key point is there is but one polypeptide. And your quaternary structure, well, that's also R groups that are going to interact, or R group interactions. But this is on 2+ peptides. So more than one peptide is going to be what stabilizes that. Now one detail that I think is important to add to my definitions for tertiary and quaternary structure is if you look at, your primary structure, we define the types of bonds. We define that it's a covalent bond, specifically the peptide bonds that stabilize that structure. This as an example, X of covalent bond. Okay. Backbone hydrogen bonds. Well, that's a non-covalent interaction. And so we have a covalent, non-covalent interaction, which are going to stabilize these things. I think that it's important to think, okay, well, what is the stronger of these two? Well, your primary structure stabilized by covalent bonds. Covalent bonds are stronger than hydrogen bonds. It's just, I think, a good reminder to keep that in perspective. Now, your tertiary structure, these are R group interactions. So as we know with the different amino acid R groups, we could have any number of different options. We can have, I'm going to say, NC or non-covalent interactions. Examples being, well, hydrogen bonds. Think about the amino acids where hydrogen bonding can take place. You could have two serines, a serine and asparagine, serine and aspartic acid. Then you could also have your covalent bonds. Well, there's one example, and that would be the cysteine bond that can be established. So two cysteine residues, what they can do is they can form what's known as a disulfide bond. And we'll get back into this in a little while, but this is one example of two amino acids forming covalent interaction with one another. There's tons of non-covalent interactions. There's hydrogen bonds, there's hydrophobic, all right, H-phobic interactions that can stabilize these. Now, in quaternary structure, it's pretty much going to be your non-covalent interactions, but there also can be that disulfide bond. It's not super common, but that can happen in fibrous proteins. So I think that it's good to think about, well, what are the types of interactions that are stabilizing these different levels, not just what are the parties that are interacting, sure, your R groups, but what types of interactions. Now, there are some conventions and kind of standards that people use. When you describe something that's 50 amino acids or less, you generally refer to that as a peptide. Whereas when something is more than 50 amino acids, it's a protein. Now, is this like a make or break? It's not really. This isn't something that I would like lose a lot of sleep over. I mean, I wouldn't think like, oh my gosh, I referred to a 49 amino acid molecule as a protein instead of a peptide. Like I said, don't lose any sleep over it. But generally, these are the conventions. Now, with that in mind, I'll refer to, let's say I'm talking about hemoglobin. I'll talk about a peptide or a region of that protein. I'll say this, 10 amino acid long peptide because it's a part of the protein. Now, most proteins contain 50 to 2,000 residues, or 50 to 2,000 amino acids. The average molecular weight for an amino acid is 110 grams per mole. Okay, so that's your molar mass. But the convention in biochemistry is to refer to, and the convention when it comes to amino acids is to refer to the molecular weight of one amino acid as 110 Daltons or 0.110 kilodaltons. The molecular weight of proteins in Daltons, or DA, 50,000 grams per mole, is 50,000 Daltons, which is the same as 50 kDa or 50 kD. These two are pretty common. And I say that just because let's make some very rough numbers or very rough estimates. If you have a protein or if you have a molecule that's 50,000 Daltons, well, let's see, how large is that? 50,000 Daltons. Is that going to be a peptide or a protein? It's going to be a protein. How large is it? Well, let's see. If it's 400 amino acids, that's 400 times 110. Okay, so we're looking at about 44,000 Daltons. So, let's come up with, we're looking at, anyway, my point is that we're looking at a protein that's about 450 amino acids. Is my math a little bit off there? Yeah, it is. But I'm in the general vicinity. So we're talking about a protein, and we're talking about something that's more than 450 amino acids long. Now, polypeptide chains are flexible, yet they can have some restriction. So if you've ever heard the phrase protein folding, It's this flexibility that people are referring to. They're saying, well, what does this thing look like? And, when we talked about hemoglobin, I alluded to the different states, the different folds, the different shapes of the protein. Now, there are some bonds within a protein or within a polypeptide that have some limitations. Now, if we look at this molecule right here, we need to look at a couple of different bonds. So if you see this molecule right here, it's important to be able to look at this and identify the peptide. Your peptide bond is this bond right here. So between carbon and nitrogen, because the convention whenever we're looking at a protein or an amino acid is we read it in the C or the N to C directionality. Okay. Now, there's these bonds that follow that peptide bond. And these bonds have different rotations. This one right here that I'm highlighting the bond, this is known as the phi angle. And this other one, indicated right here, is your psi angle. Now these two bonds, they have some flexibility. So whenever you see a polypeptide or a protein, it's important to be able to identify where your peptide bond is and where your phi and psi bonds or your phi and psi angles are. Now, in addition to that, taking a figure like this and kind of processing, visually processing it, aligning it with this, give yourself some, or be patient with yourself, excuse me, and understand what are the atoms that you're actually looking at there. Okay, so here I'm going to look down at the bottom figure. One of the things that I notice with my bottom figure here is that I have a phi and then I have a psi. So if this is a phi, that means that this blue ball is my nitrogen. This is a carbon. This is a carbonyl carbon. So C and C. Now, I always look at, so then in addition to that, this kind of gold ball is basically my space filler. That is my R group. So my amino acid R group. Now, with that in mind, I think that it's helpful to, let's go ahead and use my left hand and my right hand as this plane right here, left. So those atoms, which I'm going to do some erasing and then some X's. Erase. So I've got this, which I picked the wrong color because I'm coloring red on red. Let me switch again. Let's go with, keep going black because that's all right. I guess white would be all right. These, these, yeah, I got a circle it. And this. These atoms are basically kind of on one plane. Now, I always kind of visualize it like this is my left hand. And that is what I've just kind of highlighted here. Now, if you look at my left hand, all of those atoms are kind of on this surface right here. They make up my left hand. Now, what I want you to recognize is that my left hand can do this. But what my left hand cannot do is this. Can't do that. So just imagine that these atoms are on my left hand. Then I've got my right hand. Okay, my right hand is this plane right here. So what can happen is that I can have rotations. And whenever I have that rotation, the R group that is displaying from where my middle fingers connect with one another, well then that R group is going to rotate. Okay. Now, what's going to be able to happen is that my hands are going to reach a point where they're most comfortable with one another. they're going to be able to rotate. They're going to have some complementary angles. And so what I want you to think about this is my phi angle, which is kind of the end of my left hand, and my psi angle, which is at the closest point on my right hand. These two bond angles will rotate and they'll get to a kind of complementary pairing or a nice bond angle pair. So my phi and psi angles, those are going to be where my rotation takes place. That's going to lead to the overall change in the structure of my protein. So when you think about a protein folding, I want you to think about, okay, phi and psi angles. Those two things can rotate. Now, those phi and psi angles will rotate and they'll find a safe kind of pairing. We have what is known as a Ramachandran plot that illustrates that acceptable or that appropriate pairing of phi and psi angles. Now, if you look at the figure on the left, what you'll see, so every point on here, actually, rather than the one on the left, we'll look at the one on the right. This graph on the right shows on the y-axis our phi angle or our psi angle, and then on the x-axis our phi angles. And so this shows the favorable combinations of angles that your phi angle can be rotated to and that a psi angle can be rotated to. So if you were to look at this and say, oh, well, I have analyzed my protein and I can see that I have a phi angle that's approximately 90 degrees. Okay, so phi angle, 90 degrees right here. Okay, so then the psi angle, let's say that you ended up with, oh, the psi angle is negative 60 or negative 90. That would mean that you're right here. Now that would be an energetically unfavorable combination of phi and psi angles. So the Ramachandran plot just gives basically an inventory of those favorable phi and psi angles. Now, this all plays into your protein secondary structure. As I mentioned before, now, as I stated previously, secondary structure of your proteins. This is stabilized by backbone interactions, and these are hydrogen bond backbone interactions. So it's between a carbonyl and an amide. Now, there are three types of structures that result from this. Your predominant or your primary structures are the alpha helices and beta strands or beta sheets. Now, there are also loops and turns, and I'll mention those. But when it comes to like understanding and being able to kind of like put them together, I would recommend having a strong understanding of the alpha helix and beta strands. Here's an example of what an alpha helix looks like. Now, one thing that I want you to notice about this is that an alpha helix is a right-handed helix. Now, when we look at this structure and you hear right-handed helix, I think that it's the sort of thing that you look at and think like, neat, okay, cool, got it. But here's where it gets a little bit, I don't know if I would say complicated, but this is kind of like adapt your mind to think about it this way. So take your right hand, okay, and imagine that your thumb, the palm of your right hand is facing you. So I am looking directly at my palm. My thumb is going in the center of this alpha helix. Now, my fingers, whenever I basically close them in, they are folding towards me. Okay, so if I can draw a poorly drawn hand with fingers here, there we go. There, yeah, this looks like my ability to draw a hand really has not changed since second grade or so. So if I close my fingers in, well, then they are basically like this ribbon right here. They're closing back towards the hilt of my hand. Okay. Now, that's always kind of weird because discerning between a right-handed and a left-handed helix is almost like, well, if I just sort of changed the rules and I said it was going down, wouldn't that make it a left-handed helix? And the answer is no. The convention is that this is known as a right-handed helix. The alpha helix is a right-handed helix, and this is kind of that definition. Now, a DNA double helix is a left-handed helix, or yeah, it's a left-handed helix because it basically does the opposite. Now, there's a couple of things that I want you to notice about this helix, and I'll highlight them in a couple of different colors. So first of all, I'm going to get my highlighter out. Here, here. Here's a ring. There's a group. There we go. Let me erase my poorly drawn hand. Dun, 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 dun. Then I've got highlighter, that. Okay. So what I highlighted are the R groups of your amino acid, or R groups of each amino acid. They're projecting outward. So they are on the exterior of the cylinder, the exterior of the helix. Now, the next thing that I want to highlight, I'm going to use the highlighter tool, but I'm going to do it in, let's go with red. Yeah, there we go. This, 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 here. Where I made these red markings, those are illustrating my hydrogen bonds, my backbone hydrogen bonds. So the terminology or the explanation for this is that your hydrogen bonds, H bonds, are parallel to that axis of your alpha helix. So they're basically kind of think about the hydrogen bonds as fortifying that alpha helix in the sense that they're basically performing or they're providing like structural integrity for the alpha helix. Now, there's a couple of other things, and here's a nice little model for model A. C, we've got more of we don't have the ribbon structure, but or sorry, B, we don't have the ribbon structure, but instead we have the atomic structure. And C, we've got a view down the center of it. So we're kind of looking from here's my eye, and there we're going, that's a pupil right there. Yes, these are my eyes, and that's, yeah, we're looking down. And then here is a space filling model, model B of what that looks like. Now I'm going to toggle back and forth between this slide and this slide. I really like this model right here. I think that it shows a good depiction of an alpha helix because you have all of the different components. Sure, it's got the ribbon diagram, but that ribbon represents your alpha helix. Now, one of the things that I want you to know is if you were to look at the structure, it's kind of, if you kind of travel back in time to when you took what is it, geometry or algebra 2 or something. You talked about wavelengths, or heck, whenever you took physics or general chemistry, you talk about wavelengths. And this from here, from X to Y, is one complete wavelength. So what that means is you started from this point and then you ended at this point. This, where I just drew a circle and a line through it, and another line, that would be half of a wavelength. We have some sort of the same visual whenever we are looking at an alpha helix. I'm going to erase my brilliant modeling there. And I want you to know that from this point to this point, that is one rotation. So that is one complete rotation. There's a couple of important things you need to know about that one complete rotation. The first is that that one complete rotation has a height of approximately 5.4. I love that symbol right there. That's an angstrom. So 5.4 angstroms. That's smaller than a nanometer. The next thing about that one complete rotation is that it takes approximately 3.6 amino acids to complete that one rotation. So it's not 4, it's not 3. Instead, it's 3.6 approximately. Now, the significance of that is that amino acid residues are not directly on top of one another. So if we jump over here, this would be an R group. This too would be an R group. Notice how they're staggered. They're not right on top of one another. So that has to do with this, 3.6 amino acids per one rotation. Now, when we go down to the lower left-hand corner of this, I'm going to bring this, and I'm going to also write 5.4, I'm going to say 5.4 angstroms tall, one rotation. When we go to the lower left-hand corner, we have basically a display of the hydrogen bonding pairs. Okay, so we have a hydrogen bond that's established between a carbonyl group and an amide. Now, what this is showing us is, let's go ahead and imagine that, let's see, can't find a good starting point. Okay, we have an amide and a carbonyl group that have established a hydrogen bond. Now, if we were to look at this polypeptide, we'd say that this is amino acid 1, 2, 3, and I'm looking at basically this is 1, this is 2. And I'm going to itemize out, here's my N, C, C, 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 N, C, C. Okay, so that's amino acid 3, amino acid 4, 5, and 6. Now, what is also illustrated here is that we have another hydrogen bond. So we can look at this and we can say, okay, well, the carbonyl group from amino acid number 1 forms a hydrogen bond with the amide from amino acid number 5. So 1 and 5 hydrogen bond. Then the carbonyl from 2 and the amide from 6 hydrogen bond. Okay, now let's jump back here. That would be like this right here, 1, and this is, what was that again? 5. Okay, so then this is 2, and this is 6. So we have this network, this kind of continuing network. And there's a pattern for that. And that pattern is referred to as I plus 4. So that we can say, okay, well, there's a network of hydrogen bonds, and it's a carbonyl that at I that forms a hydrogen bond with an amide at 4, or I plus 4, I'm sorry. Now, what this means is that, let's go ahead and write out some of the hydrogen bond pairs that can arise. I'm going to erase all of this, and now hydrogen, or these bonds, the alpha helix doesn't go on for forever. So you might have 1 and 5, 2 and 6, 3 and 7, 4 and 8. Now here's where it gets kind of tricky. What comes after 4? I think it's 5. That's right, it is 5 and 9, 6 and 10. Oh, but well, this is kind of funky. But remember, the first number that's written is the carbonyl. The second is the amide. So the carbonyl of 5 hydrogen bonds, the amide of amino acid number 9. Now, when you conclude an alpha helix, you might only have, or you're going to have amino acid residues that only hydrogen bond with one amino acid or one backbone. So this might be where that alpha helix kind of concludes. 6 and 10. Okay, so then, well, does 7 have a hydrogen bond? Yeah, it does. But it only hydrogen bonds between its amide and amino acid 3's carbonyl group. Now, this is a little bit complicated to think about, but just imagine that this helix goes on, and then at some point, the helix kind of concludes. Okay, I just sneezed. All right. So the next type of secondary structure is a beta sheet or a beta strand. Now, these beta sheets or beta strands, what they do is a little bit trickier because it's not necessarily I to I plus 4. Instead, these can get fairly complicated, but they form some really crazy cool structures. If you've ever heard of the protein GFP, also known as green fluorescent protein, green fluorescent protein is a series of beta sheets that come together and they make this like barrel or cylindrical type of structure. So as we proceed here, there we go. The most important thing about beta sheets and beta strands is basically their orientation. They can form two major structures, a parallel and an anti-parallel beta sheet. Now, the anti-parallel beta sheets, we still have an amide and a carbonyl, amide and a carbonyl that establish a hydrogen bond, okay? Now, what I want you to visualize here is this whole thing right here, that thing right there. That's a polypeptide that looks like this, okay? This right here is another polypeptide, okay? We read polypeptides in the N to C directionality. Now, if you notice this arrow right here, that blue arrow, that's indicating that we're going N to C, okay? So this would be our N, and this would be our C termini. Now, this arrow indicates that, well, this peptide is going in the opposite direction. So where the C termini of my first peptide right here, that's going to align with the N termini of this second peptide right here. So this would be in the N, and this would be the C. So it's almost like we have a structure that looks a little bit like this. The N aligns with the C, and the C aligns with the N. Okay, so that's where that anti-parallel. So they're aligned with one another, so they're parallel, but they're going in opposite directions, which is where anti comes from. Now, if we compare and contrast that to our parallel beta strands, here's our N going to our C. Then we have the next peptide that is, again, the same arrow, N going to C, N going to C. Effectively, the way that that's going to happen is if you think about anti-parallel as like looping back in on itself fairly easily, a parallel beta sheet or beta strand would look something like this. In order to get the N to align with the N, the C to align with the C. Now, I'm just referring to the N as one region or one portion of a peptide. So it's not the N termini, it's an N termini of a segment. So the difference between the parallel and anti-parallel beta strands is, and this is pretty easy to detect, but it's almost like it's hidden in plain sight. If we look at these yellow dashed or these yellow boxes with the dashed black lines, what do you know about the or what do you recognize that makes the anti-parallel different from the parallel? This anti-parallel, straight up and down. parallel, kind of angled. Now, when you think about, um, if you think about like proximity and strain, because if you remember anything from organic chemistry, it's, torsional strain. That's one of the phrases that I remember. If we look at these two, this angled hydrogen bond from our parallel beta strands, these are weaker. Anti-parallel, by contrast, because they are straight up and down, they are stronger. Because they're straight up and down, that puts the hydrogen bond donor and acceptor directly across from one another, ever so slightly closer together. So it's almost like they just, by virtue of having that straight line of sight, they're stronger than one another, or they're strong. The anti-parallel are stronger than the parallel. Now, this third group of structures, these turns and loops, these are a little bit, they are organized and ordered, but they're a little bit less organized as a parallel and anti-parallel beta sheet, or sorry, than beta sheets and alpha helices. They're still relevant, they're still important, but what I want you to know is kind of that they exist and that there is a little bit of a pattern to them. Now, when it comes to a beta turn, we have something similar to that of an alpha helix. Rather than an I to I plus 4 pattern, we have an I to I plus 3, where we still have a hydrogen bond that connects those two. Now, this beta turn is something that's kind of common in a beta sheet. It's basically the turn that makes that sheet possible to form. When we look at all of our different amino acids, there's a couple of things that I want you to know. One of those things is that this amino acid right here, proline, and this amino acid right here, glycine, these two amino acid residues, more than anything else, these are secondary structure breakers. And I highlight these two. So when you think about secondary structure, there are some amino acids that are more likely to be found in an alpha helix, and there are some that are more likely to be found in a beta strand or a beta sheet. Okay. Um, there was a paper that was, or there was a review article that was put together back in the 70s by two researchers who basically took all the data that was present at the day and they came up with a nice little statistical model where they said, this residue is more likely to be found in an alpha helix. This is more likely to be found in a beta strand. Awesome. Now, these two amino acids, however, they stood out. These are secondary structure breakers. And I bring them up because they're at two opposite ends of the spectrum. Proline. If you look at proline, I always had trouble visualizing proline whenever I was an undergraduate. It wasn't until I said NCC, that's my amino acid backbone, and then I said, okay, proline has basically, I've got to connect my alpha carbon to my N termini. How am I going to do that? Well, I'm going to do it with a total of 3 carbons. So there's this little 5-member ring that I made with proline that connects my N termini to my alpha hydrogen again to my N termini, my alpha carbon. Ugh, sorry about that. So this is a proline residue. Now, what's significant about this is that if this phi angle, if that tries to move at all, well, it's basically this nitrogen, the N termini is anchored again in a separate way to my alpha carbon. So this residue, a breaker, it is a breaker because it is too rigid. Because this phi angle has a limitation on how much it can rotate. Now, glycine, by comparison, let's look at glycine. NCC, okay? We know that this is our R group, right? Yeah, we know it for a couple of reasons. One reason is it's got a different color. That hydrogen is a little bit like it's not quite rust. It's not quite brown. It's not quite maroon. So So I would say it's like yeah, I'd say maroonish. We can pick that up. But if you didn't have that color pattern, well, who's to say that it's not this hydrogen? This is the only amino acid that is not chiral. All of your other amino acids are chiral centers, or their alpha carbons are chiral centers. The R group is the exact same as one of the other groups that's on there. So because of that, that makes glycine such a small R group that it is a breaker because it is too rigid. Or not rigid. The opposite of rigid. It's too flexible. So when it comes to secondary structure, those are some of the things that I want you to know. Now, we've already looked at and classified our different amino acids. I think that this is just another great resource that you can look at to identify your amino acids and get more comfortable with them. Now, our next level of protein structure is our tertiary structure. This is stabilized by all types of weak and strong interactions. I kind of highlighted that here, here, there. So we have all types of different interactions that stabilize the tertiary structure. Now, this includes super secondary structures and domains. There are a couple of structural motifs, which if you remember our conversation about, or my lecture on hemoglobin, hemoglobin had a structural motif known as a globin fold. Okay, so that fold is a little bit more complex than these, but these structural motifs are kind of patterns that are observed in structures, in protein structures. There's a helix loop helix structure. There's a coiled coil. So this is one coil and another coil that basically wrap around one another to form a coiled coil. A helix bundle, a beta strand, alpha helix, beta strand unit, a hairpin loop, a beta meander, a Greek key, and a beta sandwich. These can get much more complicated. There's a beta barrel, which is a bunch of beta sheets or beta strands that come together to make a cylinder. Now, when it comes to domains, these are parts of proteins that independently fold, or they're independently folded or functional units. Now, these domains might do different things, or they likely do different things. For example, here is one polypeptide, one protein chain. Highlighted in orange is a portion of the protein that binds the substrate glyceraldehyde 3-phosphate. Okay, that is a key molecule in glycolysis. Then here's another part of the exact same polypeptide. And what it does is it is a dinucleotide binding domain. It binds NAD+. This is all one polypeptide. And effectively, and this is the case with many different multi-domain proteins, is you could cut these two in half, and this would be able to carry out its function. This would be able to carry out its separate function. But they kind of work together because they basically evolved to have that combined activity and combined function. Now, tertiary structure. Here's tons of different proteins, and you can kind of get lost in like, okay, what am I supposed to gather from all of this? One of the things that I want you to gather from this is that when you look at these proteins, you see some similar structures. For instance, human serum albumin. It's a bunch of alpha helices. Cytochrome B562, alpha helices within this random coil. We looked at pig retinol binding protein. We've got an alpha helix and then a bunch of beta strands. Okay, so these proteins get very complicated and very complex. But at the end of the day, there's looking at secondary structure that lead to these overall structures. Now, with that in mind, not every single part of a protein is going to be in an alpha helix or be in a beta strand or a beta sheet. Now, when we look at different amino acid R groups, which is what we see here, I want you to think about what are the types of interactions that would be found between these two. So we look at these two residues. We've got hydrocarbons. What do you think those hydrocarbons are going to want to do when water's around? That's right, they're probably going to want to repel it. Okay. Now, when we look at something like this, which if we look at this next one, that could be one of two amino acids. That could be Q for, that's right, glutamine. GLN is your three-letter code. This could also be N, asparagine, ASN. And when you look at that residue, okay, yeah, it's a polar amino acid. Then over here, we have a ring and an OH group. What do we know about that OH group? It's going to love the hydrogen bond. So what's it looking for? Well, it's looking for a hydrogen bond donor, or it's looking for an acceptor or a donor. Now, when we look at these two structures, what type of interaction is going to form between them? Hydrogen bonds. Down at the bottom, what type of interaction is going to form from a negative charge and a positive charge? That's right, an ionic interaction. Now, tertiary structures, you're also going to have some hydrophobic residues, and what they're going to do is they're going to pack well with one another. And then hydrophilic residues that are going to love water and they're going to pack well together. Quaternary structure, multiple proteins folding together to make one complex structure. That one complex basically has a singular function. Or not a singular function, sorry. That complex is one complete unit. So basically what that means is that individual components of that protein, individual subunits, make that protein incomplete. Now, protein structure stabilized through covalent bonds. Some of our weak interactions, H bonds. Ionic interactions. Hydrophobic interactions. And then we've also got van der Waals interactions. Protein structure is determined primarily by X-ray crystallography and nuclear magnetic resonance, or NMR. These two methods are the most common methods. And if I remember correctly, they do have some limitations. NMR specifically has a size limitation in that I think that it's something like 50 kilodaltons. You can't really solve the structure by NMR of a molecule or of a protein that's larger than 50 kilodaltons. X-ray crystallography doesn't have that, but there are some technical limitations to X-ray crystallography, just as there are with NMR. And then a third way that's a little bit less common, just because you have to deal with an extremely large protein, is known as cryo-EM, cryo-electromagnetic electron microscopy. Microscopy is how I always want to say it, and I also don't want to say it that way. With cryo-EM, your proteins have to be very, very large. Protein complex is what you're visualizing with this. Now, when it comes to analyzing and working with proteins are very stable. If you remember, it's the primary structure that's going to stabilize them. Your primary structure is covalent bonds, your strongest types of bond. Can you disrupt a protein? Yes. And when you do so, it's referred to as denaturation. You're denaturing a protein. You're unfolding that protein. One way that can be done is with thermal energy by heating it up. So we're talking about 100 degrees or higher. Keep in mind, this is an aqueous environment, so it's probably in water. And so what's your boiling point of water? 100 degrees Celsius. pH. Well, your protein is going to be least soluble at its pI. You can also use detergents to destabilize them. Um, Detergents are hydrophobic. Then you can use chaotropic agents like urea and guanidinium chloride. Now, these two compounds, known as are examples of chaotropic agents, and what they do is they disrupt hydrogen bonds. Then also agents or reagents that are critical for disrupting protein structure, reducing agents. Beta mercaptoethanol, also known as BME or 2-ME, and dithiothriotol, also known as DTT. These are reducing agents, and what they do is they disrupt cysteine bonds that stem from disulfide bonds. Because if you remember, a disulfide bond is one type of reaction or one type of bond that can form between two amino acids. These disulfide bonds, as covalent bonds, are strong. So you can't adjust pH and expect those bonds to break. Instead, what you have to use is a denaturing or a reducing agent like this. Now, there was a guy back in the 1950s, I believe. Maybe it was 40s. I think it was like 58, 59. His name is Christian Anfinsen. I think that he is like super cool. I'm not a structural biologist. The only classes that I took on structural biology, I was like, man, this is very, very complicated. I don't understand it. Um, but I think that what he did was really clever and was really smart, really profound. Because what he did was he said, hey, there's this protein, RNase. RNase can very easily be purified. RNase can be purified and can be very easily analyzed. And you know what I'm going to do is I want to study how this protein basically responds to a chaotropic agent like urea. So that's going to disrupt all the hydrogen bonds. So if we look at this polypeptide right here, this protein, we've got a lysine as our N-terminal residue. I should switch to the highlighter, not the eraser. There we go. Um, he said, okay, so we got this lysine here. You know what? I see, let's see, I see an arginine and a serine. You know, there's probably hydrogen bonding there. And then there's definitely hydrogen bonding between like let's see, a lysine and a tyrosine, probably between aspartic acid and asparagine. And I'm going to throw a bunch of urea at this protein. And whenever I do that, it's going to denature it. But one other thing that I'm going to add is DTT. And what I'm going to do is I'm going to see this protein go from this structure right here, this whole thing right here, to, we're going to label this as new structure. And it's going to look like what I'm drawing in this box. It's going to go from this 3D structure to basically a straight line. And, you know, everyone was like, Christian, we know that. And he's like, I know. But I think that what we're going to see is that there's a bunch of disulfide bonds that are broken, hydrogen bonds that are disrupted. This protein's going to unfold, and then you know what I'm going to do? I'm going to take this mixture that we got, um, and what I'm going to do is I'm going to get the urea, and I'm going to basically dilute that out, and then so that's going to eliminate the chaotropic agent. So the hydrogen bonds are going to recover. And then I'm going to also remove the DTT, the dithiothiotol. And so what I think is going to happen after I remove those is that this like fully unfolded protein is going to go by the wayside. And what I'm going to get is this right here. I'm going to get my protein. I think it's going to refold. And everyone was like, I mean, that's a grand idea, Chris. Can I call you Chris? Yeah, I'm going to call you Chris. But I just don't know. And Christian was like, I think that we have to do this. And he did. And then it was discovered that lo and behold, that's exactly what happened. Now, the significance of this is that this basically meant that everything that's produced in your primary structure of your protein, which that was preserved because when you added the DTT and the urea, you lose secondary and tertiary structure. Those things go toast. Those are toast. But then whenever you remove the urea and the DTT, the primary structure remains, your secondary structure recovers, and your tertiary structure recovers. So essentially what that means was that everything in the primary structure was adequate to recover your secondary and tertiary structure and then ultimately your function. Now, this is tremendous because essentially what this means is if you know the primary structure of a protein, there's a pathway or an avenue or a mechanism to figuring out the tertiary and this the tertiary structure of a protein. So this is really grand because we have tons and tons of access to genomes, which give us access to primary structures of proteins. And if we know the primary structure and the primary structure allows you to predict tertiary structure, then you'll be able to analyze a protein's function and analyze a protein shape without having to actually solve that protein structure because your primary structure was enough to predict the tertiary structure. That's what I think is so fantastic about this, because it basically said that everything that's the tertiary structure is coded in the primary structure. That's pretty good. Tertiary structure is coded in the primary structure. There we go, right there. Signed, sealed, delivered, right there. Okay, now this is a more detailed explanation of what happened. So the steps are: 1, 8 molar urea denatures the protein and beta mercaptoethanol or DTT. Cleave its disulfide bonds. So these disulfide bonds are busted up. Okay. Then removal of only the mercaptoethanol allows the disulfide bonds to reform. So protein, this is 3. Subsequent removal of the urea allows the protein to be scrambled, and then adding a small amount of the beta mercaptoethanol to the scrambled protein in the absence of O2 catalyzes its conversion to the active enzyme through disulfide interchange reactions that allow the native disulfide bonds to form. Alternatively, that's number 2 here, we have the removal of the denaturant and the reductant, the beta mercaptoethanol or DTT, allows the protein to denature and reform disulfide bonds in the presence of oxygen. So the sequence, when we remove both of them simultaneously, both your denaturant and your pleiotropic agent, your protein can go back to its desired structure. Okay, so Christian Anfinson, awesome. That's all there is to it. We've talked about the hydrophobic effect being a contributor to protein stability. And there's this plot right here, hydropathic index. This is also known as a hydropathy plot. And basically what this is, on the x-axis, this shows our amino acids from residue 0 to 240. This is our sequence, and you can classify amino acids as being hydrophobic or hydrophilic. Hydrogen bonds fine-tune the structure, and surface electrostatic interactions or salt bridges contribute very little to the overall structure. So We've got lots and lots of hydrophobic residues, and what these guys are going to do is they're going to seek out other hydrophobic residues or other hydrophobic substances within a living or within a cell. Disulfide bonds help stabilize extracellular proteins. Metal ions can also stabilize internal proteins. Zinc coordinates strongly with sulfur, nitrogen, and oxygen because they all have lone pairs of electrons. Zinc fingers are common in nucleic acid binding proteins. Now, when it comes to a protein folding, going from its denatured structure, depicted right here, to its fully native folded structure, there's a couple of basically ideas on how it's done. Levinthal's paradox is that finding the native folded state of a protein by a random search among all possible configurations can take an enormously long time. Yet proteins can fold in a second or less. So the idea here is that with the Levinthal's paradox is a denatured protein doesn't have to say, okay, well, if I put this arginine here and I put this histidine here, I'm going to try to fold to all of these different configurations in order to find the native state. If I did that, it's kind of like you're arranging your room for what would be the most comfortable positioning of all the furniture in your room. You're like, okay, if I put this here, put that there, put that there. No, that's not good. I got to try it over again. So if that were to happen, the amount of time that would take would be basically infinite. Whereas a protein is going to fold and get to its native state in a matter of seconds. Now, protein folding, there's a couple of different ideas on how it takes place. So one idea is that, or there's a couple of different theories on how it takes place. And truth be told, proteins get to their native folded state a couple of different ways. So I think that if we look at this model and we say, okay, where X and Y are, those are two fully unfolded denatured proteins. They are both going to end up right here at position Z. I'm going to say Z. They're at Z. How are they going to get there? Well, for one, we can look at this and just say, well, X is going to follow this path and it's going to arrive there. Y is going to follow this path. And if you look at this model and you say that, you're effectively correct. Now, there's a lot more beneath that. So some proteins are going to, and this is, whenever I say X and Y, this is the exact same protein. But some, I guess, copies of one protein is going to take the route of, well, I'm going to fold all my secondary structures. And then all my hydrophobic residues are going to collapse in on one another. And then my tertiary structure is going to be formed after all of that is said and done. So that's one option. An alternative is that your hydrophobic residues are going to kind of be clustered together, and then secondary structures are going to come together or be formed, and then you're going to arrive at your tertiary structure. This is basically showing the amino acids that are going into the proper position and the routes that they're going to take. Now, there are molecules known as chaperones, commonly known as molecular chaperones, and these are proteins that help proteins fold. So some examples are HSP70/HSP40. HSP is a heat shock protein, trigger factor, chaperonins, and HSP90. Now, these are protein complexes that basically take a partially folded or an unfolded protein and help it fold into the right configuration. These configurations can vary quite a bit. And you can look at one particular region or look at one particular polypeptide, and that polypeptide, depending on everything else that's in that full protein, might take on a couple of different structures. Here, what we have is this and this polypeptide, VDLLKN, forms an alpha helix right here, and then forms a beta strand right there. Now, proteins are going to be very dynamic, and they're going to take on a couple of different structures, and they're going to not only take on different structures, but this is one of the drawbacks of protein structure determination. Proteins are dynamic. They're constantly in motion. I always, and I don't know how accurate this is, but I've always theorized, maybe not theorized, but I've always thought to myself, well, if you're trying to solve the structure of a protein, that's kind of like solving the structure of a wave. And not like a wave like this, but instead like a wave crashing into the ocean, where it's like, when that wave is in this specific position, when it's, 5 feet from crashing and basically flattening out, well, that's going to look different than when it's, 4 feet, 11.9 inches from crashing. And then it's going to look different from when it's 5 foot 1 inch. These are all going to be variable. Proteins, they observe that. They exhibit that. They are constantly changing. These are in motion. Every atom within a protein is in motion. So if you solve a crystal structure, that term crystal is not by mistake. You have crystallized, you've frozen that protein in time, and you're looking at it in one specific instance or one specific instant. It might change a nanosecond to nanosecond later. Now, protein structure leads to protein function, which is kind of what we took from Christian Infantson's work, where the more you know about a protein structure, the more that you will learn about a protein's function. There are proteins like collagen, a structural protein. That's a component of skin, bone, tendons, cartilage, and teeth. Now, this is a protein that forms what's known as a triple helix. That means it's 3 polypeptides that wrap around one another, and they form a helix of helices. Glycine appears to be at every third residue, and the sequence pattern of glycine, proline, hypoxanthine, and glycine, proline are common. This right here, hypoxanthine, is it's hydroxyproline. This is a post-translationally modified form of proline. There are no intramolecular hydrogen bonds. Proline residues prevent alpha helix formation. And proline residues and resulting in left-handed helix formation, which is different from our alpha helix, which is a right-handed helix. Covalent crosslinks are formed. Now, the degree of those crosslinks increases, and with age, making meat tougher, just as an example of visual of that. Now, antibodies are also proteins that what they're able to do is they are able to detect specific epitopes, specific molecules, and they have a region that is very sensitive to that. So these variable domains and that antigen binding site, so this entire variable domain is what's going to detect one molecule different from another molecule, and that is rooted in its primary structure. So primary structure of antibodies is going to be different from one to another, and they're going to produce different products, and they're going to detect different products. All right, so when it comes to protein structure, there's quite a bit to unpack. I would recommend that you definitely, I would say before anything else, is you definitely have to have a firm grasp of your amino acid structures. You have to be able to look at the amino acid structure and say, okay, that residue and that residue will interact through a hydrogen bond. This residue and this residue will not interact. Serine and leucine, for instance, if they interact, it's not going to be strong, because that's going to be serine loves hydrogen bonding. Leucine loves a hydrophobic interaction. Chances they interact, pretty slim. If anything, it's going to be an induced dipole. But that's going to be a very, very temporary interaction. All right. Well, I hope this is helpful and I hope you have a good one.